Hi everyone, welcome back to class. I welcome everyone specially to today's class. For all my returning subscriber, thank you so much for your support. And for those that are coming across this channel for the first time, I want to say welcome. And I promise you, you will enjoy this channel. Subscribe to this channel so that I'll be able to make this video more visible to people to be able to learn from it. Thank you once again. Thank you very much for your support always. I'm grateful. So, everyone, welcome back to class. It's learning time, and today, what we want to talk about is gene regulation in prokaryotic. And the example we'll be talking about today is the lactose operon. The lactose operon, also known as the lac operon. Lactose operon is a regulatory mechanism that is found in E. coli. And what they do is that they help in regulating the expressions of genes that are involved in the metabolism of lactose. Let me take that again. They help, it's a regulatory mechanism that is found in E. coli, and their function is to help us to regulate the expressions of genes that are responsible for the metabolism of lactose. Remember, in our previous class where we talked extensively on catabolite repression, where we are talking about the process in which the cell helps to suppress the uptake of all other molecules there, as long as the carbon source that it prefers is present. And their preferred carbon source is glucose. I will leave a link to that video in the description so that you can follow and really study what catabolite repression is all about. And also, a knowledge of that will help you to understand lactose operon more. Now, let's move on to lactose operon. We said it is a mechanism that is a regulatory mechanism that helps in regulating the expressions of genes responsible for the metabolism of lactose. And what this means is, let's take for example, you want to grow a bacteria in a medium, and in that medium you provide glucose, they have lactose inside the medium there where they are, but we have little a source of glucose. On the other hand, we have enough lactose that is present. You know, when we talked about the catabolite repression, we said the cell will prefer to take up glucose because glucose is a simple sugar that can easily be metabolized by them. But in this case, although there is glucose, but we have low concentration of glucose. Let's take an illustration of that. In a medium we want to grow, in a medium of bacterium, we have bacterium here. B represents the bacterium that we're talking about. So we have it here. Now, inside those medium, we have glucose. We have glucose. Inside there, inside the medium, we have glucose. So we have glucose, we have glucose. Also, inside the medium, we have lactose. Lactose, 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 lactose. Although there is glucose, but we have little availability of glucose. Why we have lactose in abundance? So what will happen? This the cell will now sense that there is no glucose, so and I need energy. I need to metabolize. So the cell will now helps in the uptake of lactose. The cell will help in the uptake of lactose. So lactose will be used in place of glucose. But also note, lactose cannot be used directly by them. The lactose still needs to be broken down to glucose and galactose. So that is part of the reason why they prefer to uptake glucose in the first place. But since there is no glucose, they don't have a choice than to take up lactose. So back to the board, and let's say for example, this is our bacterial cell. This is the inside of the cell, then this is the outside. Now, here we have glucose, we have glucose, but on the other hand, we now have lactose. We have lactose, 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 lactose. Now, since there is not enough glucose here, the cell will sense that I don't have sufficient glucose, I need to uptake lactose. And on this cell, we have permease. Permease. The work of permease is to enhance the uptake of lactose into the cell. You know, if it were to be catabolite repression, it is the glucose transporter that will enhance the uptake of glucose. But here, permease will now enhance the uptake of lactose. So we have lactose entering into the cell. Upon the entry of lactose in the cell, we already have what we call lactose operon. We have an operon in the cell. And how does the operon and how does this operon look? In the operon, you have promoter, we have operator, and we have the three genes there. We have lag Z, lag Y, and lag A in the operon. We also have lag I. This one is also known as the repressor. The repressor. Now, for this lag operon that is here, let me give you what you can use to remember it quick. Let us put it here. P-R-O-G. Your P is the promoter, your R is the repressor, your O is the operator, your G is the genes. Now, each of these genes, what they encodes. Number one, gene LAGZ encodes beta galactosidase, meaning that as soon as there is expression of this gene, it will produce beta galactosidase. The function of beta galactosidase is to help us to break down lactose into glucose and galactose. Now, the second gene, which is LAG-Y, help us to code for permease. It codes, please follow this arrow. It codes for permease. What is the work of permease? It enhances the entry of lactose into the cell. You see the permease that is here. It is the one that will enhance the entry of lactose into the cell. Now, the third gene is lag-A. What it codes for is thiogalaktoside transacetylase. And the function of this is just to help us to sieve out every toxic thiogalaktoside that can enter. It might have entered into the cell. So you can just say that it's like a sieve. Let's go back to the operon. Repressor. We bind with this operator. As long as this repressor is binding with this operator, there is no how RNA polymerase can move here. It's like a gatekeeper. So here we have our lag-I. So which is our repressor, our active repressor now. You can say active repressor. So as long as this repressor is here, there will not be movement of RNA polymerase that is coming that want to help us to perform the transcription of these genes will not be able to move. Now, upon the entry of lactose into the cell, the lactose will now be converted into allolactose. The lactose, when lactose enter, so lactose is converted to allolactose. by beta galactosidase. This allolactose will now bind with this our active repressor. So when our allolactose binds here, I'm using A to represent our allolactose, it binds here, there will be change in the conformation of this our repressor. When there is change in the conformation of this repressor, it will not be able to bind with this operator. It's like when, let's use example, you have something like this and something like this is coming. You know easily it can fit in into each other, but as long as the conformation of this thing is changed, maybe it could be changed to this that is coming will not recognize it again. Now when there is change in the conformation of this our active repressor, and as long as the conformation is changed, it will not be able to bind this operator again, so it will be removed from the operator. So we can say it's now being dragged down, so it's unable to bind to this operator again. Upon dissociation of this our active repressor from operator, then what will happen is that RNA polymerase will be able to bind with this promoter and activate this promoter. And also remember that the combination of this our promoter with our repressor, I said, is just like a gatekeeper. Now you see that dissociation has opened the gate for us. So as soon as this gate is opened, then RNA polymerase will bind with this promoter, then initiate transcription. Then it could now move past this because the gate has already been opened. So just free entry. And as it's just initiate the transcription, now we'll now move to these genes. The laxid, we said it encodes for beta galactosidase. So as soon our operon is already active now, it is the process has commenced. So our laxid will now give us beta galactosidase. And remember, the work of beta galactosidase is to help us to break lactose that enters into the cell to glucose and galactose. So that one will help us to do that. After that, like why, which will now give us permease. Remember the function of permease to enhance the uptake of lactose into the cell. So as soon as permease is being expressed here, we have this our permease, then that means we will have more permease available. So now on the cell we now have permease, permease. So as we're having those permease, then like that, our lactose will now be entering into the cell. So we have more lactose entering into the cell since their gate has been opened also by permease. So we have it entering into the cell. Then also, lastly, lab A, which codes for galactosidase transacetylase, help us to filter every toxin that is present. Now, can you see now that when beta galactosidase is expressed, it will help us to break lactose. Then permease increases the uptake of lactose. As the uptake is increased, then beta galactosidase is helping us to break it down. Then the bacteria can now make use of the product from the breakdown, which is we get glucose and galactose. So they will not be able to make use of it for their metabolism. So I hope we all understand everything about lactose operon now. So can you see that it is not something so big or something so difficult? It is just an easy thing. As soon as this operon is turned on, then there will be metabolism of lactose. But whenever this operon is off, then the bacterial cell cannot make use of lactose. So I'm trusted that we understand everything about lactose operon now. With this, we've come to the end of today's class. Please, if you have any question, you can drop it for me in the comment section, and I promise to answer you as soon as possible. See you in the next class. Bye bye.